You are listening to Professor Megan Workman. Welcome to this lesson about viruses. In this lesson, you will learn what is a virus. You will examine its structure and relate that structure to different entry strategies of different viruses trying to get into their hosts. We will examine ways of beating a viral infection, and we will look at two different life cycles of viruses. Viruses are considered non-living because they don't meet the characteristics of living things that you learned about in a previous lesson. They're not made of cells. They cannot reproduce except with the help of their host. They don't really grow and develop. Viral genomes do evolve, however, but viruses generally don't respond to stimuli. They cannot use energy outside of their host. And they don't have a lot of complex mechanisms for regulating the internal environment separate from the outside world. So viruses don't meet many of these characteristics of living things. They are absolutely dependent on their host cell to do most anything. Here's a simple cartoon rendering of a virus, which is just genes in a box. Viruses are minimalist. They have a protective coat called a capsid that's made up of proteins, and inside that, the thing that they're protecting is their genome, which could either be RNA or DNA. And this contains the viral genetic code, the instructions that the virus is going to use when it takes over the host cell. Viruses are so minimal that this limits its capabilities. That means that each virus can only infect a narrow range of hosts, such that different viruses infect plants or bacteria or animals. Even within an organism, sometimes viruses can only infect certain tissue types. Like within humans, there are certain viruses that can infect epithelial cells only, but cannot infect muscle cells. And this, again, has to do with the minimal genetic code that limits the capabilities of the virus. Some viruses have evolved an additional structural component called an envelope. And the envelope is made up of something similar to a cell membrane. And it usually contains glycoprotein spikes that serve a function to promote viral entry into the host. This is an evolved level of complexity that enables these enveloped viruses to enter hosts more easily than non-enveloped viruses can. Here's an example of an enveloped virus. We have the same nucleic acid surrounded by the protective capsid in the middle, but here we've just added this viral envelope around the outside. Let's compare and contrast enveloped viruses versus non-enveloped viruses. Here in their cartoon renderings, you can see that they both have genetic material enclosed by a capsid, but on the enveloped virus side, the capsid is then also surrounded by this membrane envelope. So structurally, that's the difference. Enveloped viruses have an envelope, non-enveloped viruses don't. The envelope is decorated with glycoprotein spikes, which are not present in non-enveloped viruses because they lack the structure of the envelope that the spikes reside in. Enveloped viruses with these spikes can fool host cells by mimicking healthy molecules that are normally imported through the cell membrane of the host cell. So the function of these glycoprotein spikes of enveloped viruses is to mimic something that the host cell naturally spends its own energy to import. These spike proteins allow docking, and usually docking on the cell membrane, it's called docking because it's like a boat docking. Um, the virus can travel up to the cell, it connects at a docking site, and there's some either straight up chemical bonding or some affiliation, some attraction chemically between the molecules of the viral envelope and the cell membrane. And this is influenced heavily by the pH of that environment outside of the cell. But once these spike proteins dock, then the cell membrane imports the virus. That contrasts non-enveloped viruses because they don't have those spike proteins. They have to sneak in through other means. And how they do that is they get up close to the cell, and when the cell is feeding, it's importing food and other molecules on purpose, and the virus just kind of has to hide among some food and sneak in that way. So it's imported quite on accident, these non-enveloped viruses. Because of their differences in structure, they have different resistance to fluctuations in the environment. Non-enveloped viruses actually tend to be more resistant to changes in temperature. They are more resistant to some chemical agents that, like disinfectants. And non-enveloped viruses are more resilient generally. And that's because the envelope itself of enveloped viruses can be easily damaged. It is broken open by certain chemical agents like soaps. It can be easily damaged by heat. These glycoprotein spikes can be malformed, in which case the virus would have a more difficult time entering the host cell. Just to give you some examples of each, both viruses exist. There's many examples that infect humans. Enveloped viruses include SARS-CoV-2, which is the virus that causes COVID-19 disease. HIV is an enveloped virus. Influenza viruses are enveloped. Contrast that with examples of non-enveloped viruses that include polio, noroviruses. Many of both types of viruses can infect human tissues. Because of the current pandemic, I thought it would be interesting to show you a cartoon rendering of SARS-CoV-2. Here you can see the spike proteins, which you've probably heard about in media by now. SARS-CoV-2 has two types of spike proteins that are both involved in viral entry into host cells. The S1 spike chemically attracts a receptor called the ACE2 receptor on your cells, and this is what allows the virus to dock on your cell membrane. Then the second spike protein, S2, actually forms a structure connecting the viral envelope to your cell membranes. The combination of these interactions of spikes S1 and S2 with your cell membranes allows the SARS-CoV-2 to inject its genetic material into your cells in order to infect you. Here I've circled a receptor binding domain, so this is the part of the spike protein that's actually chemically attracted to the ACE2 receptor on your host cells. Kind of interesting to think about a real life example that is salient right now. So here's a micrograph, or a picture taken through a microscope of a virus-infected cell. You can see the nucleus of the host cell, and then all those little things are viruses in production. And what's happening here is that the host cell is becoming filled with viruses. All of its cellular processes have been taken over by instructions from the viral genome. And so the host cell's own organelles for producing proteins and replicating DNA or RNA. All of these cellular processes are taken over by the virus so that your cellular machinery turns against you and becomes virus producing factories. Hundreds of new viruses are going to be produced until this host cell bursts open, releasing all those baby viruses into the body. Each one can then go on to infect another cell. So it's this cascading infection. Gross. So how do you beat a virus? That's your immune system's job, to go around searching for infected cells. And infected cells are noticeable based on the structure of their cell membranes, and your immune system is sensitive to that and can then attack infected cells, effectively shutting down those viruses that are inside and being produced. But your immune system, if it's coming in contact with a novel virus for the first time, it may not yet recognize that that is something it needs to attack. That's why vaccines are sometimes used to train the immune system with just the part of the virus that the immune system needs to learn to recognize, so that if it happens to see that again, it already knows to attack it, and you're not wasting any time training your immune system. And vaccines work. Another way to beat a viral infection is to prevent it from getting into your cells in the first place. And there are pharmaceuticals like Truvada, if you've heard of PrEP, that act like, well, it acts like a condom for your cells that prevent viruses from getting in. And PrEP is really effective at preventing HIV virus from being able to dock with host cells. Therefore, a person could be exposed to HIV, but if they were taking PrEP, the HIV would not be able to infect their cells. So despite exposure, you would not become HIV positive if things are working correctly. The third way to beat a virus is to take a pharmaceutical, RX here standing for prescription, that prevents the viral genome from replicating. So certain antivirals are designed to do this. One is called acyclovir, that treats herpes infections like shingles or cold sores, and it mimics viral nucleic acid, but it's chemically different from true viral nucleic acid. So then when it gets incorporated into the viral DNA, it shuts down DNA replication. So it's an ingenious way to prevent the virus from replicating within the host cell. So here I wanted to give a cartoon rendering of what the immune system might look like when it's attacking a virus. So these are called antibodies, this blue and red upside down Y looking thing. And what the antibodies do, these are produced by your immune system. And you have an individual antibody for each individual pathogen that you have ever been exposed to. As long as you have immune memory of such a thing, you can still produce these antibodies. So these antibodies circulate around, usually in varying numbers. There might not be a whole lot of antibodies at any given time, but it doesn't require very many, because what they do is that Y-shaped part has a binding affinity to the spike protein here on this enveloped virus, for example, and the antibody sticks to it and acts like a flag, waving over immune cells that will then attack and kill whatever the antibody is attached to. So the antibody could attach maybe to the virus itself, but more frequently to infected cells that are also producing or have these spike proteins on the cell membrane due to being infected with the virus. So the antibodies flag over the immune system to destroy the infected cell. And your body can ramp up antibody production once you're infected or with the help of vaccines. Another treatment for COVID-19 was to take monoclonal antibodies. So this would be antibodies that were previously produced by an infected person. You inject them into someone who's recently gotten sick, so they're not able yet to produce their own antibodies. But once those antibodies exist in the bloodstream, they can attach to the infected cell, waving over the immune system cells that can kill infected, antibody tagged cells, and that is an effective way to clear out a viral infection as well. Next, let's talk about two viral life cycles. One is called lytic and the other is lysogenic. All viruses have a lytic cycle, but some can have a lysogenic cycle. And let me break these two down. So a lysogenic life cycle of a virus is called the long game. It's when viral DNA hides within the host cell genome, such that every time the host cell divides, it reproduces, viral DNA gets copied into the daughter cells. And this could produce no symptoms. You're just accumulating more and more cells in your body with this viral DNA that's hiding. And you might not have symptoms until something triggers the virus to become active and switch into its lytic life cycle. Now, I call the lytic life cycle the short game. This is a golf reference. Short game refers to putting. It's when the viral DNA or RNA starts coding for the production of baby viruses. Your host cell becomes the viral production factory. You become ill, you have symptoms, and all these little baby viruses escape into circulation and try to infect more and more cells. So the lytic phase is the one that makes you feel sick. The lysogenic phase is when the virus is hidden, it's not causing symptoms, it's hanging out until something triggers it to go lytic. So all viruses have a lytic phase. This is when your immune system can find them and attack them. But when the viruses, sneaky viruses that can go lysogenic, they're hiding in your genome and they're not causing any alterations that might inspire your immune system to attack those infected cells. So some lysogenic capable viruses include herpes, HPV, HIV, things that can infect a person long term, that are not completely curable, but you may not experience any symptoms for long stretches of time. To review what you have learned in this lesson about viruses, you have learned that viruses are non-living minimalists that have a genome and a protective protein capsid. Some viruses are enveloped with an additional structural component called a membrane envelope that functions to aid viral entry into host cells. That's how SARS-CoV-2 works. The other types of viruses are non-enveloped, meaning they just have the genome and a capsid, and these are more limited. They have to get into the host cell in a more sneaky way. We talked about three ways to beat a virus. You kill infected cells, you prevent entry into the host in the first place, or you can inhibit viral genome replication within the host cell. Viruses can have two different life cycles. The lytic one, which is the short game in which the host cell becomes a virus producing factory and the host organism feels symptoms, versus the lysogenic phase, which is the long game where the virus embeds itself in the host genome and it hides there for perhaps years at a time, during which the host is infected with the virus but may not experience symptoms.